All right, the lymphatic system, or the lymphoid system as your textbook calls it, is all about defending against disease and any type of pathogenic substance. Basically, this is your immune system. And when I was in college, I took several anatomy and physiology classes, human-based and animal-based, and for some reason, I was always just bored to tears when the instructors got to the lymphatic system. And then one day I took in grad school an immunology class, and it was an entire class, an entire semester devoted to the immune response and how our body fights off pathogens and fights off diseases. And I found it to be one of the most fascinating, most exciting classes that I've ever taken in undergrad or grad school. And I really enjoyed it. So hopefully you guys will get some enjoyment out of this as well, and not find it as boring as I initially found the lymphatic system when I was in college. So if we look at some of the anatomies of the lymphatic system, of course we've got lymph nodes that we'll talk about here in just a moment, and some other lymphatic organs, but we also have lymphatic vessels. And we're going to find these lymphatic vessels running alongside blood vessels, because the point of the lymphatic system and the lymphatic vessels is to take fluid out of the tissues, put it into these vessels, and then take it to a lymph node or some other lymphatic organ, clean the fluid up, put it back into the bloodstream. So you're going to see these lymphatic capillaries, these lymphatic vessels kind of running alongside blood vessels. One place, however, you're not going to find lymphatic vessels will be the central nervous system. And we kind of alluded to that a little bit earlier when we talked about the nervous system, and particularly the microglial cells. We can't send pathogens directly to the central nervous system. It's just too risky to do that. So, and that's the job of the lymphatic vessels, to carry fluid that could potentially contain pathogens to lymph nodes so that we can eradicate those pathogens. So we start with lymphatic capillaries. And unlike blood capillaries, the lymphatic capillaries kind of have a blind end to them. So it's not a true circulation here. We're going to take fluid out of the tissue. It's going to enter the capillaries, at which point it becomes lymph. Lymph is just tissue fluid that's in the lymphatic capillaries. It can get in pretty easily, but it can't get out as easily. And then the lymphatic capillaries are going to all converge into lymphatic vessels. Lymphatic vessels superficially resemble veins because they're thinner walled, like veins are, and they have valves in them to assure a one-way flow of fluid, like the veins do. The lymphatic vessels then converge into the lymphatic trunks. The lymphatic trunks will then converge into one or two collecting ducts. We have the thoracic duct that's going to drain the left side of the head, left side of the neck, left thoracic region, and everything beneath the diaphragm. Okay. The thoracic duct arises from the cisterna chyli, which is an enlargement in the abdominal cavity that really counts as the beginning of the thoracic duct. And then we have the fluid that doesn't enter the thoracic duct. It's going to enter the right lymphatic duct. That's going to drain everything on the right side of the head, neck, right side of the thorax, right arm, but not the right leg. Remember, anything below the diaphragm, below the rib cage, is going to be drained by the thoracic duct. When we discuss the organs of the lymphatic system, we talk about them in terms of primary lymphatic organs and secondary lymphatic organs. The primary lymphatic organs are those that actually produce the cells involved with immune responses. The secondary lymphatic organs are going to be those organs that are important for immune responses but do not produce the cells themselves. So, along that token, the primary lymphatic organs are going to be the bone marrow, particularly the red bone marrow, because that's, of course, where most of the white blood cells are going to be produced. And then the thymus gland. The thymus gland, again, is where we're going to mature and allow T lymphocytes to develop. And remember, the T in T lymphocyte stands for thymus. And we talked about the thymus gland when we talked about the endocrine system. Now, the really cool thing here is we got to allow these cells to develop from these hemocytoblasts, and then we, if it's going to become a T cell, we send it from the bone marrow to the thymus, and then it matures in the thymus. If it's going to be any of the other types of white blood cells, it just remains in the bone marrow and develops into whatever type of white blood cell it's destined to become. But we have this concept of immunocompetence. We have to do two things with these cells as they're developing. First, we have to make sure that they do not recognize what we call self-antigens. Okay? All of our cells have glycoproteins, glycolipids on the surface of it that serve as self-antigens. And your antigens are different than my antigens. Everybody's antigens are different. That's what makes us unique individuals. So the first thing we do as the B cell or the T cell is developing within their respective places is we introduce a cell to them that has a self-antigen on it. If that developing B cell or T cell or any white blood cell, but if that developing cell recognizes and binds to that self-antigen, we kill that cell. Because when cells recognize antigens and bind to them, then ultimately what's going to happen is you're going to destroy the cell that's bearing that antigen. So we don't want them to pay attention to our cells. We don't want them to bind to our own cells and destroy them, because that gets into something called an autoimmune disorder we'll talk about later. But if they pass that first hurdle, they don't recognize, they don't bind, they don't destroy the cell with the self-antigen, we allow them to continue to develop further, and then right before we're ready to release them out into the bloodstream, we introduce a foreign antigen to them, a non-self antigen. In this scenario, we want them to bind to that cell presenting that antigen. We want them to destroy the cell with that non-self antigen on it, because that would mean that it could recognize something foreign that shouldn't be there. So if it doesn't bind to recognize and destroy the non-self antigen in the cell presenting it, then we kill the cell, because we don't want to put all the energy into developing a cell that's going to get out into the bloodstream, out into the tissues, that can't recognize foreign substances and pathogenic substances. Okay. And that's what immunocompetence is all about. It means that your immune cells will recognize foreign substances in the body, but they should not recognize and act upon your own cells and your own antigens. The secondary lymphatic organs, the lymph nodes, play a big role, of course, in immune responses. We'll talk about those here in just a moment. And then the spleen, which is really just kind of one giant lymph node. So if we look at this graphic that's presented here, you of course see the secondary lymphatic organs and the one of the primary lymphatic organs with the thymus gland. But you look at the distribution of lymph nodes, and that's what I like about this graphic, and that's what its point is to show you, is where these major lymph nodes are. So you've got cervical lymph nodes in your neck. You may have felt a swelling behind your neck. We've got some behind our ear too that can swell. When we have an infection in the armpit, in the groin, those are often common sites of swelling in the lymph node when we get an infection as well too. If we look at a lymph node, so we always see these nice little cartoony graphics that an artist has put together. It makes it look like a really interesting organ, but when we look at a lymph node from a gross anatomical standpoint, it's just a mass of tissue really. But you've got this capsule that surrounds it. The capsule invaginate inward to create these trabeculae, and then the trabeculae create the follicles within the lymph node. And in those lymphoid follicles, we're going to have B cells. We may have some T cells in there. There's going to be a cortex. There's going to be a medulla associated with this as well too. And again, a cortex is just an outer portion of an organ. A medulla is an inner portion of the organ. But you're going to see vessels coming to the lymph node, and you're going to see vessels leaving the lymph node. So all these little guys here that are bringing dirty fluid, dirty fluid to the lymph node, those are the afferent vessels. Remember, when we talk about afferent versus efferent, we talked about with the nervous system. Afferent's going to bring something to something. Efferent's going to take something away from something. So, afferent vessels bring the dirty fluid to the lymph node. Then you're going to have efferent vessels leaving the lymph node at a little indentation called the hilum, and they're going to be carrying clean fluid. Okay? And you're always going to have more afferent vessels than you do efferent vessels, and that is to create a bottleneck effect. Okay. If we had, so in this case we've got 5 afferent vessels coming in here. If you have 5 afferent vessels coming in and 5 efferent vessels going out, fluid would come in, go out at the right rate. They're at the same rate. And you would have no time to filter that fluid. You'd have no time. Okay, in these lymphatic nodules, and these, we'll also call them germinal centers here. In these germinal centers, you've got B cells. At this medullary area, you've got T cells. You've got macrophages in these germinal centers. You've got to give them time to actually find, bind, and engulf and destroy the pathogens. And if the fluid's coming in and going out at an equivalent rate, then that's not going to happen. So we have several afferent vessels bringing fluid to the lymph node. Okay. We slow it down because we don't have that many efferent vessels leaving the lymph node. And while that fluid is kind of hanging out in the center regions there, in the germinal centers there, the B cells, the T cells, the macrophages, the neutrophils, whatever they are, can all do what they need to do to clear out that pathogen. Okay. If there's a pathogen in that lymphatic fluid that's in that lymph node, you're going to get a swelling of the lymph node because you're going to start producing more of an appropriate cell type to go through and combat that infection. The spleen is just like one big old lymphatic lymph node. Okay? It's composed of red pulp tissue. That's where the red blood cells are going to go to die. Remember, we said it's the erythrocyte graveyard. You've got white pulp tissue. White pulp tissue is going to be composed of lymphocytes, white blood cells, B cells, T cells, around parts of what we call the splenic artery, blood supply to the spleen. But it's going to go through and it's going to clear up any pathogens. Again, it functions like a really big lymph node in addition to being the site where red blood cells are going to die. Your spleen is not necessary. It's not vital for life. You get rid of it. Your liver can do much of the same things that the spleen can with regard to red blood cell destruction and recycling. And then you've got, of course, other lymph nodes that can take over the lymphatic function for that. Some other lymphatic organs we have are the lymphoid tissues. And you've got your tonsils, which are basically just clusters of lymphatic tissue in the mucous membranes of the pharynx. You've got your palatine tonsils. So you look in the back of the mouth. So let's say this is your pharynx back here. Here's your uvula that hangs down in the back of your throat. You've got these muscular arches on either side of the pharynx, and kind of in a little pouch in here are the palatine tonsils. To kind of, provide an extra layer of protection as we're swallowing foods. Tonsillectomies used to be an everyday occurrence growing up in kids. If you get your tonsils infected, you get tonsillitis, several times, they just go through and take out your tonsils. Tonsillectomies aren't as necessary nowadays because we have more effective antibiotics. But those are the palatine tonsils. You have the pharyngeal tonsil, which is kind of posterior, right behind the nasal cavity where the nose opens into the throat, a region called the nasal pharynx, often referred to as the adenoid gland. So if you hear somebody talk about the adenoid gland, it's that pharyngeal tonsil. And then you have the lingual tonsils located near the base of the tongue. And again, they're just lymphatic tissues that contain lymphocytes and provide a little bit layer of protection in the oral cavity. Then we have MALT and GALT. MALT is mucosa-associated lymphatic tissue. GALT is gut-associated lymphatic tissue. These are just little nodules of lymphatic tissue scattered throughout the mucous membranes of the respiratory system, in the case of MALT, and parts of the reproductive tracts, and then also scattered throughout the digestive tract if we're talking about GALT. Okay. And then you have Peyer's patches. Peyer's patches are also lymphatic nodules, but they're a cluster of lymphatic nodules located specifically where the small intestines meets the large intestines, where you get that junction there. You're going to find Peyer's patches. And again, just an extra layer of protection, because what's in your digestive tract, whether it's stomach, oral cavity, small intestines, large intestines, rectum, whatever it is technically outside your body, so it's an exposed area. So we're going to put down these extra lymphatic tissues to potentially eradicate anything that could get in and cause a problem.